Okay, hi. So let's welcome back to the second practical session. So today what we are doing is continuing from our first practical session, which we have identified our microarray data into 4 different types. So you can treat it as group stage 1, stage 2, stage 3 to stage 4 cancer. So what we are going to do now is, can we find stage 1 specific genes? That means genes that is overexpressed only in stage 1, genes that's overexpressed only in stage 2, stage 3, and stage 4. And what do they mean? So at the end of the day, we are going to use this thing called your gene ontology enrichment analysis. So you're actually looking at the subtypes of genes, or rather the stage of cancer, which will increase the expression of certain genes, and then what are the biological functions they have increased. So for example, it can be metalloproteinases, it can be extracellular matrix, degradation of extracellular matrix, and so on. So without further ado, let's start with our R session. So let me move this part. So we go to our R studio. Okay. So if you still remember your R studio, the first thing that we need to do is to change the directory. Okay, let me just change the directory. Oops. So I go to sessions, I set working directory. So I go to, for example, I'm using my desktop, practical, and practical 2. Okay, so open. So what I have at this moment is, this is my practical 2. And obviously, I have the workspace from the end of the first practical, which is the end of day 1.r data. Okay. So we go to our document. Now what we want to load up is a few set of libraries. We want to load in Lima, we want to load in the microarray data, databases, and so on. So let's load up these few libraries first. If you are doing it successfully, you should not get an error. Okay, so let's run through. Okay, so at this stage, I need you to go and scan through. Are there anything that's error? So attaching looks all right. Okay, following, nothing give me the big word error anywhere. Okay, so everything is looking well. So then let's load up the data from last week. Okay, this x.r data is the workspace from last week. So we do not need that. We need groups and filter expression. This is our first microarray data. Remember? From our last week's week 1 practical. So we have our R data. So let's load it up. Okay. So just double click and then you load into the global environment. Say yes. So then what we have is, we have all the data that starts to pop up. Okay. So this is why we end last week. We want the filter expression data as well. So the filter expression, let's load it up. Okay, it should be loaded in the first place already. And we load up the groups data. Okay, so let's load this up as well. Okay, all right, so all the data looks all right for now. Then what we want to do is we look at our silhouette plot from last week. So we can run the silhouette plot. Remember, this is what we have. So we have the silhouette plot from last week. So let's run the command and let's see what happens. Okay. So this is our silhouette plot, if you remember correctly. Now, what we want to find out, what we want to do is to remove these 2 negative silhouettes. Okay, these 2, let me go to zoom. So I want to remove these 2 negative silhouettes. So negative silhouettes means that the width is actually in the negative range. Okay, so let's go. So how do I remove this? Basically, I set a remove whereby I'm in the 3rd column, whereby it is less than 0. So the width is less than 0. So I put it under a variable called remove. Then I call out remove to actually see what happens. Okay. So it gives me the data. So it is basically sample number 8 and sample number 51. So this is sample 8 and sample 51. Okay, we can't really tell. Then what we want to do is we want to filter it. Okay. So our filtered expression, our original 62 microarray data, or 62 samples microarray data. We want to filter it off, remove the 2 negative silhouettes, and we end up with only 60. And we put it in a variable called filtered.expression. We also remove the groups as well. Because remember, the groups is group A1, group 2, group 3, group 4. So you want to remove both at the same time so that these two are in sync with each other. Okay, so that's all, nothing very much. Okay, then if you look at here, so your filter expression data, you remember that our original microarray data set, each sample have 1,500 probes. So we remove two samples, we actually remove 3,000 probes in all, or 3,000 data points. So we left with 90,000 from 93,000. Okay. Yep. So that's what we have. Then this is where we can start to analyze. We are actually now able to look at differentially expressed genes. So what we want is we want to generate a matrix, a design matrix. And what we want to do is to test for the differentially expressed genes. Okay. So we are using a linear model to fit the design in. You can actually read up a bit more about this model matrix. How does it work? Okay. So let's run this too. We have group 1, group 2, group 3, and group 4. Okay. So this is our design. We have the model matrix. Okay, you probably have to read up this model matrix and actually see what does it do in order to get more idea. But what we are trying to do is put it into 4 groups. Okay. Then we use a linear model to fit, basically a regression model, to fit the data into these 4 groups. Okay. So LM fit is a linear model fit. Okay. So then we have this fitted data. Okay, it's just a class. There's nothing much you can do about it. So again, remember what we are doing now is a sequence of steps. So some of it may not be really obvious, okay, unless you are familiar with R yourself. Okay. But we kind of treat this whole practical session or this series of practical session as kind of a recipe for you to keep doing things. Okay. The next step is to find out group 1 versus group 2, group 1 versus group 3, group 1 versus group 4, group 2 to 4, 3. So we want to find out all the contrast. So G12 means it's group 1 gene minus group 2 gene. So we want to make all the contrasts. Then we want to see the contrast matrix. Okay. Okay, so let's run that. This is quite fast. Okay. So this tells us the different contrasts. Okay. So group 1, contrast 1, 2 means that it is group 1 minus group 2, 1, 3. So we check each row, are we talking about the correct contrast? So this gives us everything. Okay. Group 1 versus group 2, group 1 versus group 3, group 1 versus group 4, then group 2 versus group 3, group 2 versus group 4, and then group 3, group 2, this is the last one, group 3 versus group 4. So we set up all these contrasts to actually see, in order to prepare for the next step. After which, what we want is to compare what are the genes that is expressed that is different from group 1 versus group 2. But in order to do that, we have to use the data from group 3 and group 4 to estimate the variability of each probe. So you use the data from all the probes to estimate the variance. Okay. So in this case, we use a contrast fit command. Okay. And we have a fitted. So we actually combine the two things. Number one is we combine the linear regression model into the contrast matrix itself. Okay. And then we use eBase to do a group fit. Okay. So let's just run this. Okay. It doesn't make a lot of sense, but unless you, again, I say that this is like a recipe for you to work with. So now build the contrast and build the groups. Okay. So all of them is fitted accordingly. Okay. What you have to remember is one is called fit and the other one is called fits, the one with the S or without S. Okay. Then the next one is we want to see a silhouette plot. For example, we want to silhouette plot based on coefficient 2. So what is coefficient 2? Coefficient 2 is the second one, the second contrast. This coefficient 1, 2, G12 is coefficient 1, G13 is coefficient 2, and so on. Okay, so it based on these coefficients to see the silhouette plot. Okay, so what we have is to generate a results table first. Okay, so we generate a results table for group 1 versus group 3. Okay, just keep the results. Now you have 1,500 observations of 6 variables. Doesn't matter. Okay, after which you design a silhouette plot based on, you only want to color it red if the log fold changes is more than 3 and the negative log p-value is, or rather the adjusted p-value is less than 0.0001. Okay, otherwise you label it gray. So we only want to see which one is red, the red probes and the gray probes. Then we plot it up into a silhouette plot based on the x-axis, which is the log fold changes. The y-axis is the minus log of the p-value. Okay, so let's run this step. And we should generate a volcano plot. So this is how a volcano plot looks like. Now, let me zoom it out. So a volcano plot, you are actually just drawing a line here based on the threefold changes, log threefold changes, and the p-value is here. So basically, the red dots tells you that they are differentially expressed genes. The gray dots tells you that they are not differentially expressed genes, or probes in this case. Okay. So if you want to increase the stringency, let me just reduce it. If you want to increase the stringency, all you have to do is to increase the log fold changes by changing this number, or to drop down the p-value maybe to 0.00001. Okay, then you will actually increase this line from here, the horizontal line from here to maybe up here. Okay, so, and we also know that silhouette plot is somewhat symmetrical to fold changes of 0, but it's not perfectly symmetrical. Why? Because when you compare between two different groups of sample, it's not likely to have exactly the same number of genes that is down-regulated or up-regulated. Okay. So it's just somewhat symmetrical. Okay. Now, having said that, let's continue. So what we want to do is, can the table actually just show us, give me the top table of, so the top 10 genes for coefficient 1. Coefficient 1 is group 1 versus group 2. Okay. So let's look at the just 10 data points, the 10 most significant probes, or most differentially expressed. Oops, I kind of messed up here. Running copy. Okay. So this is what it gives me. Okay, let me just move this aside. Okay, hopefully, let me just repeat this command again. Now you can see this table in a clearer form. So these are the top 10 most significant probes based on the log fold changes, the average expression, the t statistic, the p-value, the adjusted p-value. Okay, so these are the adjusted p-value. So this column, the adjusted p-value, and the log fold changes is plotted onto the silhouette plot. So you can also, if I want to, so these are the top 10 probes that is differentially expressed, or between group 1 versus group 2. So I can have group 1 versus group 3 by changing the coefficient. So this is group 1 versus group 3. So you can start to look at more things. Or if I want to look at, instead of the top 10 probes, I want to look at the top 20 probes. I just change the number to 20. Oops, sorry, I have S there. So I just change the number to 20. Okay, so you have 20 now. So you can basically see the top 10, 1, 2, 3, 4, 5, 6, 7, 8, 10 is the same up to here. Then the other 10. So you can keep playing around with it. You can actually generate all the data that you want. Okay, if you are comfortable with it, you can even take this data out and generate and plot it in Excel. It's up to you. Okay, so we have this. So let's move here a bit closer. Okay, it tells you what is the column. Log4 changes. LC is a log4 change between group 1 and group 2. Average expression is the mean expression between the two groups. Okay, so the average expression between the two groups, the T statistics, the p-value from the T statistic, then the adjusted p-value, which is based on multiple statistical tests. Okay, so it adjusts the p-value after multiple tests, and B is the log of the odds of that gene being differentially expressed. Okay. So what we want to do is, can I just know the number? How many genes? I just ask, okay, between group 1 and group 3, how many genes are showing 5-fold changes? 4-fold, 3-fold, 2-fold, and 1-fold. So I use this command. Okay. Now, it basically asks me coefficient 1, so between group 1 and group 2. Okay, how many genes are, n row means I count the number of rows. Okay, LFC is log4 changes equals to 5-fold changes or more. So I have 321 out of 1,500. Okay, out of 1,500 probes, there is 5-fold changes, 4-fold, 3-fold, 2-fold, and even 1-fold change. Okay, if you really want to do that. What happens if you have no fold change? 1,500, that's what you expect, correct? You can even play around and say that I want to look for genes that is at least 2.5-fold changes. It can work as well. Okay, so 3-fold change, 2-fold change, 3-fold is 709, 2-fold is 983, but 2.5-fold is 817. So you can, this is the way that you can play this data, and then you can do a plot yourself. How many fold changes, the percentage of the genes. Okay. So let's continue. We want to look at all the genes in the probe set. Okay. So we fit it into a probe set. So our probe set list. Then what we do is we want to take the probe set list, merge it with our microarray database to get the gene symbol. Because let's say for this probe, what on earth this probe measures, which gene it measures, we do not know. We want to get the gene symbol out of it. Okay, so the next few steps, we get a gene symbol, and then we merge between the probe list and the gene symbol. Okay, this is very useful. After that, we write the overall results into a table file called overall results.txt. Okay, so we have 3 commands to do that. I get my probe list, then from the probe list and my human 133 plus 2 microarray table, I get the gene symbol, I combine it and write the table out. So I have a overall results.txt. Okay. And what it writes is the overall results.txt, which is in my practical 2 overall results.txt. If I open it up, let me just open it up in a nicer way. So you see, you have probe 1, G1, G2, G3, G4, and then what we want is the gene symbol. So here, see here, the last one, these are all the gene symbols, which is then quite useful. Now we actually know that these 2 probes actually uses the are actually detecting the same gene. Correct? Okay, so we can use this type of data. So let's continue. Okay, what we want to do is then maybe want to print out the most significant probes, okay, for the top 10 most significant probes. So we just print it out into a PDF. Okay, so this is just a function to do that. Nothing very much. Just run it. Okay, where am I? Here. Okay, so I'm looking for a file called most significant probes.pdf. Okay, close this, go to my, and here I have this most significant probes. So I open it up, which is a what do you call this, a PDF file, and look at what are the top 10 most significant probes. So for example, this is group 1, group 2, group 3, group 4. Okay, so between group 1, this gene PDE11A, you can look at it, is highly expressed in group 2 versus the rest. Okay, how about next one? Also group 2 versus the rest. Okay, so you can keep looking. This is just a kind of diagnostic table data for you to look at. If you want to print more than 10, you can print 20, you can print 30. Okay, so let me just show you. So each there's 10 pages. Okay, page 1 from 1 of 10. So if I were to repeat this and I change this from 1 to, let's say, 14, or 1 to, let's say, 24. Okay. Okay, then I will have 24. See, now it's 24 pages. So I can print out, you know, basically, if I really want, I can print out all 1,500 if I really, really, really want to. Okay, but it doesn't make a lot of sense. Maybe if you want to look at all the data, okay, just print out 1,000 of them. Okay, I have no idea why do you want to do that. It's just 1,000 pages. Okay, you generate, take a long time. I think I probably should not have done this, right? Yeah, but you get where am I going? Okay, so let me kill this. I can't even kill it. Oops. No jobs, terminate jobs. No jobs are running. Now it's done already, luckily. So I go to 1,000 pages. Okay, do you want to go and look through all the 1,000 pages? Yeah, you can look for every single gene. Okay. Right, these are all the gene symbols. So you can look at, okay, this gene, you know, if you have a specific gene, for example, this, it seems to be expressed in group 2, fair bit of expression in group 4, and so on. Okay, some genes is okay. So this has very low expression in group 2, but some expression in 1, 3, and 4. Okay, yeah, I mean, we look at this. It's quite interesting, right? How does all the genes differentially expressed in every of the 4 conditions? Actually, what I mean is 4 cancer subtypes. Okay. So you can actually play around with it, and then we'll just see. Okay. So these are good diagnostic tools that you can use. Okay. You can just print out. In fact, 1,000 pages just give you 4.4 megabytes. So it's still not too bad. Okay. So after you look at PDF, why not we have some plots that do not have a gene name associated with it. Okay. For example, now it will be hard for me to find. Okay. Yeah, okay, let's see. See, this doesn't have a gene name because some of the probes do not tag to a specific gene name. So sometimes it may not be very useful. Okay. So what we want to do is to store all the gene lists for future use. So we create a list called gene list, and then store it. This gene list will come handy. We want to say what are all the genes that is overexpressed in stage 1 versus stage 2, 3, and 4. So we group stage 2, 3, and 4 versus stage 1. So you are almost like using a t-test to do it. Then we want to group stage 2, group stage 2 tumor versus stage 1, 3, and 4. Stage 3 tumor versus stage 1, 2, and 4. Stage 4 tumor versus stage 1, 2, and 3. So this is the next part, what we're going to do. Test for group 1 versus others. So test for group 1 versus others. Others means group 2, 3, and 4. Makes sense, right? Okay. So what we are trying to do is to do that, and then we are repeating the whole process all over again, okay, just by looking at the coefficient, the first coefficient, okay, and we look at the first 10 genes. Okay, so we will do that, after which, let's look at the top 10 results first. So this is actually what we have done earlier. Let's look at the top 10 results. Okay, so these are top 10 results. Now, what you have is you have a gene symbol now. Okay, remember, just remember this. 2276, 227646. So let's go up. See, we have where's my 2276? That doesn't matter. Okay, I've lost it. Okay, but we look at all the different genes, okay, with the gene symbols now. Then we can collect the gene symbols for further processing. Okay. So what we want is to generate which are all the genes that has threefold changes and a p-value of less than 0.01. Okay. So let's run this for some nice graphics first. Let's set the graphic component. Okay. Then we look at the genes. Okay. So we do a select which one of in this group 1 versus the rest. There's more than threefold changes and the p-value is less than 0.01. And we use the length to find out how many genes. Okay. So it gives us 251 genes, or probes to be exact. Or, well, we call it gene probes. Okay. So 251. Remember that because all these are labeled as probes to gene symbol, can there be duplicates? Possible. So it may not 251, it may be much lesser. So we want to look at the gene symbols for these genes first. Okay, and then we want to remove, let's say we map the gene symbols onto it. Okay, so the length of the genes is still 251. Okay, but we want to remove all the duplicates, so we use a unique function. Okay. So we remove all duplicates, and what we end up with is, only 174 genes. But this is also very interesting. It means that all the probes that do not have a gene symbol label will be collapsed into one, and that is purely rubbish. So we want to remove that as well. Okay, so we want to remove all those that doesn't have a gene symbol, and we see how many are there left. Okay, it's supposed to only give us 173 because there's only one gene that has is basically one unlabeled gene where all the probes will be thrown inside. So it's 173. So it means that we have 173 genes that is differentially expressed in group 1 versus group 2, group 3, stage 1 versus stage 2, stage 3, stage 4 tumor. Okay. We want to find out, so this is gene list, 173 genes. How are you going to analyze that? The useful case is, can we actually find some biological process for these 173 genes, which we can have. We just write into a file called group 1 genes, okay, and then we just copy it out. We just print it out, the genes into group 1 genes. So we have here, group 1 genes. Okay, so let's go to our table. So now we have this file called group 1 genes. Okay, see group 1 genes, group 1 genes. Okay, so group 1 genes, this is the gene list for stage 1 tumor versus the rest, differentially expressed in, or rather overexpressed in stage 1 tumor versus the rest. Okay. Why is it overexpressed? Because it says group 1 minus others is must be more than 3. So it's overexpressed in group 1 versus 2, 3, and 4. So it's overexpressed, or rather this 173 genes has high expression in stage 1 tumor versus stage 2, 3, and 4 tumor. So why? What functions do they have? Okay, we want to look at these functions. But while we are at it, we can actually look at some other plots. Okay, so we can actually generate a few plots to look at. So we generate a box plot. Oops, box plot to here. So this is a series of this one on plots. Okay, so we have group. You are actually using these three, group 2, group 3, group 4, as one whole set of genes, or one whole sample versus group 1. Okay. And we can also print out group 1 specific genes. I can only print out, let's say print out 10. You know, I can effectively print out all 173. You should know how to do that. Change this number. Okay. So let me just print out the top 10 first. Oops, one more here. So now we have the group 1 specific probes. Okay, so now I can open it up and see. This is the first one that we see, you know, and so on. Okay, so this can be quite interesting. You can print out for every single thing. So after that, since we have the genes, we actually store it in the gene set for the first column. Okay. So we throw the genes into 1. Okay. So the first column contains group 1. Then after that, we can do the same for group 2. So group 1, 2 versus the rest. And then we repeat everything all over again just by changing this group 2. Okay. Remove them, go into group 2, plot it out, and our data go into group 2. Okay. So instead of just now is, you see, if I do this, okay. See, instead of the first column, I put in the second column. So I can repeat this whole thing, you know, nothing very much, can plot everything together, and I can do even for group 3 versus the rest and group 4 versus the rest. So it's the same thing. I just change the matrix. Okay. So now group 4, and see, I put in group 4. So I can redo this whole set from 1 to 2, 3, and 4 together. Okay, I don't have to keep repeating it. So I do from group 2 until group 4. Okay, until here, this part. Running. No. Okay. So if everything goes well inside my, now I have group 2 genes, group 3 genes, group 4 genes, and then it's probes. That's all the data that I need. After which, I want to draw a Venn diagram. Now, Venn diagram for 4 groups, 4 group Venn diagram is actually not easy to draw, but I can still do it. Okay. Use a Venn diagram to actually draw how many overlaps there are. I do a Venn diagram. See, this is a useful Venn diagram. So group 1 have 31 specific, and then all this. So this can give me a very good understanding of the dynamics of the gene, which gene are overexpressed and so on. Okay. So again, I can save this file if I want to. Okay. Now the next step is to find functional annotations. Okay. So I will not, in class, I probably will show you how to use David, but we'll use a simpler tool this time. Okay. So let me get to okay, here. So I go to this website called Gene Ontology. Okay, find a gene ontology. I go to gene ontology resources. Okay, geneontology.org. And what I can do is using this tool called Panther. Okay, you can go and read Panther. Okay, read a bit more about Panther. What does it do? How can you use it? You can even go to the paper on Panther. It will tell you a lot more. Okay, so let's use Panther. I find Panther to be easier to use. So I want to, let's say, group 1 genes. Okay, group 1 genes. Copy the whole thing and go to Panther. I throw my gene ID here. I want to find biological process. Remember, we have three subsections of gene ontology: biological process, molecular functions, cellular component. Okay, so because my data is Homo sapiens, you have a whole list of animals here, but my data is Homo sapiens. Okay, so I just launch it. Okay, then you have this table. Okay, it only shows us the FDR, the false discovery rate p-value that is less than 0.05. So these are groups. You can see it's colored based on sections. Okay. So you can actually then look at it, negative regulation of chemotaxis. Okay, so it is that means this function is enriched exon midline exon guidance, because remember, our tumor is actually a brain tumor, so exon is actually quite important. Okay, so there seems to have some exon activity increase, olfactory bulb, interneuron differentiation. Okay, there's something to do with the smell, forebrain tumor development. You have your G-coupled protein pathway, synapse reorganization. So continue seeing, okay, you have neuroblast proliferation. So stage 1 is characteristics of the cell starts to increase faster. And we have cerebral cortex cell migration. Okay, then positive regulation of exonogenesis. Maybe the neurons are actually trying to grow as well, growing faster. Okay, synapse reorganization and all these processes. So it can be quite interesting, increase in cell adhesion molecules as well. So we can continue working on this, and then you can go and analyze for, let's say, stage 2 tumor. So what happened in stage 2? Okay. Now stage 2 probably have a few other processes. Stage 2 now seems to be okay. You have neurofilament assembly. Okay, you have fasciculation of motor and axon, restylation, you have branching. Okay, now it seems that the nerves are growing even faster. Okay, ureter morphogenesis is starting. Okay, synapse assembly. So these are all treated as though they are helping words to help you in trying to figure out, explain the biological process that happens from stage 1 to stage 2, stage 2 to stage 3. So here, see, you have forebrain reorganization and so on. So these are the words for you to construct your story. And that is, as far as possible, what bioinformatics can tell you. You have genes, group them into functions. Okay, so that is really the end of it. Bioinformatics can only bring you up to so far. Group into functions, and then you kind of have to sit down. Okay, these are all the These are all the biological process that is increased in stage 1 tumor. These are all the biological process that increase in stage 2 tumor, in stage 3 tumor, in stage 4 tumor. Then I have to link stage 1, what is happening to the cells? Going to stage 2, what is happening to the cells? Going to stage 3, what is happening to the cells? And stage 4, what is happening to the cells? And that brings me a complete picture of what may be happening when a healthy tissue goes into tumor, stage 1, from stage 1 to stage 2, and it starts to build up a story. The etiology of the cancer or the progression of cancer can now be explained. So that is really the end of this whole practical session. We are able to deduce what genes are expressed, which genes are overexpressed, enriched, what biological functions are enriched. Okay, so that's all for today. Have fun.